right there. It's in the board.
Yeah.
It's in the board.
Oh, yeah, thank you. So these first two are normal cells in what's called interphase. All right? They just have a nucleus. It's dark in color. You don't really see anything. You just see, on yours, you're going to see two little dots. It'll look pink. Now, if we go into this one, you notice how the DNA looks different? Like, what, you see more what here? Well, the dots are stronger. You see some light colored areas, right? Some light areas. So it looks more like a worm. So if you see some wormy areas that look like worms, then no, they're not worms, but that's kind of what they look like. Okay? Those are going to be prophase. So the next three are going to be prophase. Oh, Fred, I need you to put your phone away and pay attention. You're going to the lab. Okay? So those are prophase. Now we get down here. Those clearly look different now, right? Those chromosomes, like long worms, and they're kind of bunched in the middle of the cell. Those are going to be our metaphase. And then we know after metaphase, they pull apart. So those worms are going to be kind of pulled apart. And you can see in 8 and 9, anaphase, early anaphase and then late anaphase. And then telophase, we can see those new nuclei forming. The chromosomes still look like little worms. There's some white areas in there. You can still see some of the structure of the chromosomes remaining. Okay? So let's go ahead and see if we can identify some of these. Can somebody label these as we identify them on the screen? Any volunteers? Francisco. All right. Thanks, Francisco. So let's start at the very top left.
Mister, for the thingy.
Let's start.
Oh, okay.
Let's start with the normal ones. These right here are what it looks like normally. Okay, so these two are interphase. So Francisco, go ahead and label these two interphase. I just label them. Put an I on top. So I there and I there. Okay. Now let's go back up to the top. Do the chromosomes look different here?
Yeah.
Yeah, you can see they kind of have that wormy appearance, but they're still kind of in the middle of the circle. So what phase would this be? Not metaphase. Not anaphase. Not telophase. Prophase. Nice. So put a P right there. On the left. Let's look up here. So this is what you're going to be seeing, okay? So I need everybody's eyes. Anybody who's interested in doing the laboratory, I need your eyes up here. All right, so this is what you're going to be seeing. Now, go to the next one. Those worms. What would you think this one is?
Anaphase.
Well, they haven't been separated yet.
Oh, it's still prophase then.
Lined up in the middle.
Early metaphase.
So put an M right there.
M.
M for metaphase. This next one is kind of hard to see, so just cross that out. If you can't really see it very well, just skip it. Ooh, we see this pretty well. Does it look normal?
Yeah.
Yeah.
Interphase.
Interphase, I, and then I here.
More I.
And that looks like another interphase, doesn't it?
Yeah. This one would be.
What about this one?
This one would be anaphase.
Anaphase, that's a clear anaphase, isn't it? A, interphase, interphase, interphase. Ooh. I can see some worms here.
Prophase.
That'd be prophase. Prophase. Interphase.
Prophase.
Prophase. Yeah.
Anaphase. Oh wait, no. Wait, which ones?
They're kind of missing. They kind of, see those interphase? Okay. Yeah. They look a little different than up here, but we'll just call them interphase. That's okay. Now, they might look, there's never a perfect, some might look like they're in between. So just do your best and label them how, whatever you choose, you and your partner or yourself choose. You and your partner might have the same thing, might be a little different, that's okay, no big deal. Why'd you cross these out?
Oh, because they're blurry. You said if they're blurry, not to do them.
Oh, well, I think we can see, it looks like they're starting to build a cell wall in the middle, and it looks like.
Oh, those are together?
This looks like telophase.
Yeah, that looks like
This
telophase.
looks like a T, yeah. So put a T
Sorry, I thought they
right
were
there.
separate.
They're not quite two cells yet, that's it.
Hmm.
But it could be, Francisco is right, it could be they're just not focused right. So he has a great point. So you might count those as interphase, you might say that's telophase. Interphase, interphase, interphase. I'm just going to point this out. What about this one?
Which one?
Anaphase. What about this one?
That'd be prophase, wouldn't it?
Prophase. What about this one? Interphase. This one.
Which one?
This one.
Prophase?
Prophase. Now we need to get to the how you're going to collect your data. You see those data tables? You guys are going to make tally marks. Okay? I'll take that back now. Thank you.
Okay.
Thank you. So, yes. Now, it's important when you do your observations, you want to go along those lines. Don't cherry pick and just count the ones like we kind of did at the end. You want to go in a row. Okay? So, an example of that would be, so on your paper you have interphase, prophase, metaphase, anaphase, and telophase. So starting at the top, prophase, I'll put a tally mark, then metaphase, and then interphase, I had 3, anaphase, 1, 2, 3 interphases, a prophase, interphase, 2 interphases, a prophase, and then we had 1, 2, 3, 4 interphases. So 1, 2, 3, 4, and then a telophase, and then 3 interphases. 1, 2, 3. So see how I'm keeping track of that with my tally marks? And I want you to go row by row. Okay? Now, this is something that your phone will actually really help you with in the lab. Because you can take a picture, then on your phone you can actually annotate. You can actually write, make a mark on there, actually to do it just like I did on this screen. If you want, you don't have to. You can just use the microscope view, or you can, annotate on your phone. So this is how you write down, you do all your tally marks for your data. Now yours will look like this. This is what you'll see under high power. Okay, is that harder to see?
Yeah.
But you can still pick them out. So let's choose, maybe we'll start here. That looks like prophase to me. And then interphase, inter.
You gotta count the whole row?
Yeah, count the whole row. And then go back. These kind of look like either telophase or prophase. So count them either way. Inter. Oh, that might be prophase. Might be prophase. Inter. Oh, here. That looks like anaphase. See how they're kind of being pulled apart? That's like a prophase. Ooh, what does that look like? Metaphase. Okay. So it's going to be difficult to see, but on your phone you can take a picture, make it bigger. Eduardo, you want to do the lab? I mean, maybe you don't. Maybe you want to catch up on some videos, and I respect that. So this is a nice up close. This actually is a picture from that microscope right there. Pretty cool stuff. Check out. That one looks amazing. What's that? Anaphase. What's that? That looks like metaphase. Hey. All right. Right now, everything is already destiny focused for you. All right. It's all ready to go. So all you have to do is turn the light on and then look, and it'll be there. Okay? Take your picture and start counting. If you want to do advanced moves, if you want to move the slide around to look for more interesting areas, remember, remember how you move the slide with that dial on the bottom, okay? All right, if you have a lab paper, head on back and let's get started. You can go solo if you want or pair up to 3 per microscope. This is what you're going to see. Oh yeah, take your diagrams back with you. That'll help. Take your diagrams.